Introducing the ez-gSEAL 100B pressure controller.
Patch clamping is known for its difficulty. It can take months to master the skill to control the pressure for making seals and breaking in.
We have developed the ez-gSEAL pressure controller to help make seals. The pressure controller has been used on many cell types, and its success rate is as good or better than what a good patch clamper can reach. Best of all, no training is required. The pressure controller comes with a simple configuration. A tube connects the pressure output to the patch pipette, and the computer software controls pressure through a USB cable. No air tank is required. The pressure controller comes with control software. It is the same software used for controlling NeoBiosystems' automated patch clamp system. The pressure tab of the control software has 3 sections. On the left is the standard pressure control section, in the middle is the seal section, and the right section defines a train of pulses for breaking in. When a value is entered into the set box, the pressure immediately reaches the set value, as shown in the pressure box. Clicking the 0 button will set the pressure to 0. When a value, such as 10 mm of mercury, is entered into the added box, each time the plus mmHg button is clicked, the set value increases 10 mm of mercury. Each time the minus mmHg button is clicked, the set value decreases 10 mm of mercury. You may also generate a pulse using the software. For example, if you enter 100 mm of mercury in the peak box and 500 milliseconds in the duration box, when you click the pulse button, a 500 milliseconds pulse will be generated. The preset parameters are most often used for making seals. Typically for patch clamp experiments, P1 is set to the positive pressure used to keep the pipette from clogging, P2 is set to the negative pressure for making seals, and P3 is set to a small negative pressure to keep the seal stable during recording. Before the pipette enters the solution, click the P1 button to set the positive pressure. For brain slices, P1 is usually 50 mm of mercury. For culture cells, the value can be lower. After the pipette touches the cell membrane, click on the P2 button. For almost all cell types, minus 40 mm of mercury works well. Because switching from positive pressure to negative pressure is fast, a gigaohm seal can form quickly after the P2 button is clicked. Finally, a click on the P3 button will set the pressure to the small holding pressure after the desired resistance is reached.
Here is a video of a real experiment using our pressure controller. P1 was already clicked before the start of this video. When P2 was clicked, the seal quickly formed. When the resistance reached 1 gigaohm, P3 was clicked.
The boxes under the manual pressure ramp button are the parameters to define a train of pulses, as shown in the image below. For breaking in, you may define a train of pulses that gets bigger and bigger, or a long pulse that slowly ramps up the pressure. Click on the manual pressure ramp button to start the pressure train or ramp. When the negative pressure reaches a level that breaks into the cell membrane, click the P1 button to stop the pressure.
Here is an example of breaking into the cell membrane. The third negative pulse, or minus 110 mm of mercury, broke into the cell membrane.
If a DAC board is added to measure the resistance and capacitance, the process of making seals and breaking in can be automatic. The Seal tab in the pressure regulation dialog box defines the parameters for making seals, and the Whole Cell tab defines the parameters for breaking in. Clicking on the play button will start the process, and the process ends after breaking into the cell membrane. This video introduced how to use the pressure controller for patch clamping. There are many other applications, such as pressure injection, collecting single-cell aRNA for sequencing, and studying the mechanical properties of a cell. For more information, please visit our website at www.neobiosystems.com.